So if you're still with me for part 5 of our lecture, it's going to be very short. The last topic is called biochemical pathways. We've been talking about individual reactions that involve enzymes. So a substrate, which is a reactant molecule, goes into the active site of an enzyme. The enzyme pushes and pulls on that reactant, causes the reaction to happen, and then the products are released. But it turns out that in cells, these enzymes act in a sequence. So you have, as you can see on the screen, what we call biochemical pathways. And what biochemical pathways are really just a sequence of enzymes that act on a starting substrate. And then the starting substrate here, the orange circle, goes into the active site of the first enzyme. And that enzyme converts it into the product, which is a yellow triangle. But that product is then the substrate for the next enzyme in the sequence. So the next enzyme, the product of the first reaction, becomes the substrate for the next reaction. And so in that reaction, the triangle is then converted by enzyme 2 into a square. Obviously, they're using these shapes just to show you that there's a change. And then the square goes into enzyme 3, and the square gets converted into the green oval. And then the green oval goes into enzyme 4 active site and gets converted into whatever the final product is, this purple hexagon. And there don't have to be just 4 steps. There can be 3 steps, there can be 10 steps, but there's particular sequences of reactions that happen in cells, and each one of them has a very starting substrate and a very final product, but multiple enzymes acting in a certain order to create a final product. So these are called biochemical pathways. And that's really how the cell organizes the metabolism, the sequential building of something or breakdown of something. So biochemical pathways, the collection of enzymes needed to perform a particular kind of pathway, will be contained within particular compartments in the cell. So respiration is going to be a really good example of this. There's actually a sequence of enzymes that are found in the cytosol. And then there's several sequences that happen inside the mitochondrion. And so by having these enzymes located in particular places in the cell, it controls where the pathways are able to take place. So the enzymes are located in the whatever space that the reactions are needing to take place. So sequences can be short, like this example, or really quite long. And what substrate is involved in what product you get depends on which pathway you're talking about. So there are particular ones that you will learn about. We'll do some in chapter 7 and 8. And then if you go on into, say, biochemistry coursework, you would see a lot more of those. Now, when you have a biochemical pathway, and these are, in this diagram, there's a very short one that they're showing. It's just enzyme 1, enzyme 2, enzyme 3. It doesn't matter how long the pathway is. What tends to be true is that the end product, or it can be sometimes one of the intermediate products, the end product of the pathway, though, often can act as an inhibitor, a non-competitive inhibitor, towards one of the enzymes in the pathway. It can be the final product acts as an inhibitor to the first enzyme, or it can be a final product acts, sometimes it's the second enzyme, but first or second enzyme is pretty common as the target. And what this does, let's say a cell is making this green oval. And it keeps making it and it keeps making it, but at some point it's got a whole pile of green ovals. Like it's not using it as quickly as it's making it. So what has evolved is a system where the end product, when it starts to pile up, and only when it starts to pile up, some of the green ovals will then act as inhibitors towards one of the enzymes. Now, what I wish they had shown here was that enzyme 1 always had a niche on the back end here. It's not like it was always there. But when the end product, when the green ovals start to pile up, some of the excess of these end products then attaches to that binding site, which is not the active site. And when that happens, it changes the shape of the enzyme, and therefore the active site's shape no longer accepts the true substrate. And that's going to be a temporary condition. So it's one of those types of inhibition that it's not permanent. And when the pile of green ovals starts to get used up and there's not as much anymore, then what will happen is the green oval that was blocking enzyme 1 will then be released, and the enzyme can go back to its regular shape and start working again. And so this acts very much like a thermostat in the sense that when something piles up, it shuts the system down. But then when the products are no longer piled up, they start to get used, and there's not as many, then the system starts back up again. And this is called feedback inhibition. So just like in your house, when you turn on your air conditioner, it cools, and the temperature, the coolness fills the house, and it causes the thermometer to go down, which turns the air conditioner off. But then as heat creeps into your house, it turns it back on again. And so you're constantly, like your thermostat is always keeping the house within a range. It turns on when it needs to. It turns off when it has reached its point, and then it turns on and off again to keep the house within a certain temperature. In the same way, these enzymes are being turned on and off by the end product in order to keep the cell within a range of production, so that they're not making too much of the green oval, but that they're not making too little of it as well. And so the system keeps flipping back and forth between diagram A and diagram B, back and forth to keep the cell from making too much or too little of the final product. So that's called feedback inhibition. It's a very common mechanism of control of these biochemical pathways. And so that is it for chapter 6. I know there's a lot. I hope you took detailed notes. Get a lot of practice with the post quizzes.